Okay, guys, we're going to start talking about cellular transport. So, first thing we want to talk about is Brownian movement. What is that? That's kind of what is the motive force behind a lot of the passive diffusion where things move downhill. And basically, they've got a very long description here, but basically, this is it. These molecules bump into each other, and when they bump into each other, they push each other. They bump and then they careen off. The more molecules you have or atoms in a certain area, the more they're going to bombard into each other and the more they're going to move toward areas and stay where areas are less dense. That's why things move down the concentration gradient from high concentration to low. And I think we discussed this in the lecture when we talked about chemistry, that this is going to increase with an increase in temperature. So the hotter it is, the more energy these molecules have, the faster and harder they're going to bump into each other, and the faster and easier they're going to move. Differential selective permeability. Some things can come through and some things can't. And we talked about this on the plasma membrane. Draw a little schematic right here, guys. Anything that has a charge of any kind is going to have a hard time getting through here. So it's selectively permeable. Things that are lipid soluble, that are neutral, don't have a charge, they usually can go right through. This is going to keep our proteins in the cell where we need them. Proteins are normally charged, and some of the waste products are going to pass out. Now, as far as the transport, passive and active, I think we already hit on this in the other PowerPoint. Passive is just concentration gradients or pressure differences that are going to move these guys. Diffusion, if I got a barrier here, say whatever, say these guys can move through here. I've got more here than I do there, and because of Brownian motion, one of these guys is going to move, and then I'll have two on each side, and they'll be equal. But you can see there's a larger concentration gradient here going to a lower. And that's how diffusion works. The driving force, same thing we've talked about, the kinetic energy due to the Brownian movement. Solutions, we did talk about this in the chemistry. A solution, the solute is what's being dissolved, and it's what you have the least of. The solvent is what's dissolving, and it's what you have the most of. So if you have more of this than you do of that, then this will be the solvent. The solution is what happens when they're both together. There's your solute and the solvent, and you see them, and because of Brownian movement, these guys are going to space out, they're going to diffuse through there. If you've ever dropped a drop of ink into a glass of water, you will, as you watch, you can see the ink gradually filling all the entire volume of that container, and that's why. So the solvent right there, you got 100 grams, here you got 10. So you're going to have 110 grams, but since you have more of this than you do that, that's the solvent. Osmosis. Okay. Let me draw my little thing again. Now, let's say in this instance that this barrier, this guy cannot go through. So there's a problem. I have more of this over on this side than I do this side. But these particles are not able to move to correct it. What's going to happen, assuming this is permeable, I'm actually going to get water that's going to move out here until the concentration is equal. All osmosis is diffusion of water. When does water move? When the particles can't. That's it, guys. Because if you think about it, if I have more particles here than I do here, then that means I've got less water here than I do here, because more of the space is taken up by particles and less of water. So it's just diffusion of water. And of course, it's going to be opposite to the concentration of the solutes. Here's a good illustration. There's way more of this sugar on this side than there are that side. The sugar can't move, so the water is going to move over here until they have equal amounts. Not only this, check this out, guys. I will actually defy the laws of gravity by doing this. Look how many more. See, we got 3, 5, 7 over here. I don't know how many we got over there. But this membrane here, this barrier, the particles can't move. Water can. Water's going to move even if it has to go, and so to speak, defy the laws of gravity. It's not really, but it will be higher on this side than it on that side. Even though this barrier is permeable to water, and these guys are just showing the same thing. In this one, the particles can move and the water moves. So what's going to happen is we're going to get an even distribution of particles, and the water's level is going to stay the same. In this one, we've got more particles here than we do here, but this guy is only permeable to the water, so the water is going to move just like it did in the other one. Passive transport. Well, simple diffusion. If it's lipid soluble or very small, it'll go right through. Carrier mediated, facilitated diffusion. The key thing on here, guys, is this is still diffusion. It's still moving downhill. So these guys can't go through the lipid membrane. What happens is they bind to this guy, and when they bind, he turns, or he changes shape and opens out this way where they drop in. But they're still going in. The reason I'm not using ATP to force it, these guys are rolling downhill. They're not going uphill. So there's the carrier mediated. Channel mediated, we talked about those. These guys are going to have the right charge and the right size, and they're able to move in, and they are going downhill as well. Osmosis, there are things called aquaporins, but a lot of this water can just go straight through, even though it's charged because it's so small. Tonicity, should have talked to you a little bit about that on the other page. We have isotonic, hypertonic, and hypotonic. What does isotonic mean? It means there's the same amount on the inside of the cell as there is on the outside. If there's the same amount of solutes in and out, then what's water going to do? Nothing. It's not going to move. There's no difference in water concentration. Now, what if I put this red cell in something that is way more, say 3% salt solution? Well, normally our stuff is 0.9%. Sorry guys, I know you can't see that on that red background. So what's going to happen? Well, things on the inside of the red cell cannot move. So the only thing that can happen is that water has to leave. When water leaves, it's going to shrink, and that's called crenation. Okay, what if I put something in hypotonic? So again, the inside of our cells is 0.9. What if that is pure water? What if there's no salt or anything in it? then this is going to have more solutes, and so the water is going to have to go in. It will flow in, and eventually it'll pop, and that's hemolysis or lysis in general, not talking specifically about red cells. Filtration. Sometimes water and solute can be forced through by just pressure, hydrostatic. This is going to separate the large from the small particles, and the filtrate depends on the pressure. That's how our kidneys work, guys. That's how the kidney works. That's how these capillaries work. The blood pressure, which is a hydrostatic pressure, is higher on the inside than the pressure out here. So it's going to push things like blood sugar and O2 out. When we get over here, the net pressure pushing in is higher than the pressure pushing out. So then we're going to push things into the capillary like waste products. And there's the example of a filter where you have small openings, which many things are too big for, but some things can pass through. So in our kidneys, we talk about this in anatomy and physiology too. Our kidneys have filtration slits that are too big for the cells in our blood or for the big proteins to go out. But it is small enough for all the waste products. All right, guys, that's got it for cell transport. Again, ask me any questions when after you've read it. Thanks.